All right, it is right around 1 p.m. With that, I would like to welcome everybody today to our Badger Dairy Insights series. Today's topic is silage quality, from pricing to feed behavior. My name is Ashley Olson. I am the agriculture educator in Vernon County, Wisconsin. Along with myself, I'd like to introduce our work group who has worked on this series today. Matt Lippert, who is the agent in Wood and Clark counties, Jackie McCarville, myself, Matt Akins, who is an extension specialist, and Luiz Ferraretto, who is an extension dairy specialist. Right now, Badger Dairy Insights is running bi-weekly every other Tuesday from 1 to 2:30. Today, we have our program. There will not be one in 2 weeks, as we will be having the Wisconsin Dairy and Beef Wellbeing Conference. So that is something that you can come to in person over in Manitowoc or join virtually. And there will be more information about that later. So with that, I'd like to introduce Dr. Luiz Ferraretto, who is originally from Brazil, where he earned his BS in animal science from Sao Paulo State University in 2008. Immediately after the completion of his bachelor's degree, he joined the University of Wisconsin-Madison for an internship, followed by obtaining his master's degree and PhD in dairy science with a focus on applied dairy nutrition and forage quality. After completion of his PhD, Luiz joined the William H. Minor Agriculture Research Institute as a postdoctoral research associate. From 2016 to 2020, he was an assistant professor of livestock nutrition at the University of Florida. He joined UW-Madison in May of 2020. His research and extension interests are applied ruminant nutrition and management. His program is focused on understanding and improving starch and fiber utilization by dairy cows, corn silage and high moisture corn quality and digestibility, the use of alternative byproducts as feed ingredients, supplementation of feed additives to lactating cows, and the development of on-farm and laboratory techniques for forage and feed analysis. With that, we're excited to have Luiz on the program today.
Thank you. I appreciate the introduction. I appreciate the opportunity of being part of the group and discussing a little bit of what we have been learning with our research, as well as research of some of our colleagues. And today we will be discussing the topic, connecting the dots, forage quality, nutritional management, and feeding behavior. So our objectives will be first to review what are the effects of forage quality and nutritional management on performance and feeding behavior, as well as to discuss what are the potential effects of feeding behavior on lactation performance. So in order to do that, we'll start discussing a little bit about fiber intake as well as fiber digestibility. So if we consider a lactating dairy cow consuming approximately 60 pounds of dry matter per day, it's not uncommon for this cow to be consuming around 15 pounds of fiber. Right? So after fiber reaches the rumen for digestion, however, there is a lot of variation, right? So if we use literature values, we could assume that this cow will digest approximately 60 pounds of NDF on a daily basis, but within their cohorts, basically, you will find cows that may digest 9.5 pounds of NDF a day, or cows that may digest 2.5 pounds of NDF per day, right? And there are a lot of factors that contribute to that, right? Could be forage quality effects, that are animal effects that contribute to that, right? There are nutritional factors, right? So there are a lot of things that may be going on farm that may affect this animal, not only digestion process, but consequently performance, right? So if we assume that 1 pound of digestible fiber can translate into approximately 3 pounds of milk, there is a lot of milk that may be affected here, right? So if we consider some of the most important fiber quality parameters, Here's a short summary of those put together by Dr. Shaver. And basically, these are the factors that we consider when we are evaluating not only diets for lactating dairy cows, but also different forages, right? So these metrics here, they are related to whole plant corn silage. And basically what we have here are some of the parameters that were used for many years and some of the parameters that were recently included either by commercial laboratories or by nutritional models of diet formulation. So we're going to go through a little bit to discuss each of those, right? So obviously NDF is a well-established measurement that we perform to quantify fiber, right? And together with that, we have lignin. Lignin is the indigestible portion of the plant. And basically, it was used for many years by different nutritional models to predict intake. Actually, the new NRC that was just released also still includes lignin to predict energy value of diets, right? The new NRC does not predict energy value of feeds anymore, only of diets, and lignin is still there, right? More recently, a new assay called undigested NDF at 240 hours was developed and actually well implemented by commercial laboratories as well as different softwares to formulate diets. And basically, this fraction resembles lignin, right? It's not exactly the same thing because the indigestible portion of NDF accounts for lignin, and lignin cross-linking to other fractions of the NDF, hemicellulose or cellulose. So basically, UNDF will be a higher value than lignin, but both represent exactly the same thing, the indigestible portion of the fiber, right? After that, we have two digestibility assays. The first one is the in vitro NDF digestibility at 30 hours. Here we are using the 30 hours, but many researchers that work with hybrid performance trials utilize 48 hours. The new NRC actually has 48 hours as the standard NDFD instead of 30 hours. And, you know, it could be any number as far as you measure that consistently, right? And then there is the TT-NDFD, which is a method that was developed by Dr. Dave Combs here at UW-Madison, and it stands for Total Tract NDF Digestibility. It's not actually the total tract we just discussed on the previous slide, which is the total tract digestibility of dairy cow, but it's a predicted value after running 3 time points of in vitro incubation, 24, 30, and 48 hours, as well as an indigestible NDF assay like the UNDF 240. Okay. So basically, when we think about forage and diet quality, what we want to do with those numbers is to reduce lignin or UNDF and to increase NDFD or TT-NDFD, right? So obviously, there are many practices that you can do that. We'll be happy to answer questions about those if you have any, but that's not the topic of our presentation. Today, we want to show if you are successful in changing those numbers, regardless of the practice that you're trying to do, what does that mean to the dairy cow? So thinking about the dairy cow, right, we often consider those numbers because the fiber portion of the diet has very high potential to limit intake because of rumen fuel, right? Indigestible fiber gets stuck in the rumen, and then consequently, because you're reducing the passage rate, there is a limitation in space for the cow to continue to consume, right? When we are talking about high-producing dairy cows, this is actually a big deal, right? If you think again about the new NRC now has some equations to help predict intake based on the diet, right? Based on the forage portion of the diet, based on the lack of digestibility of forage of the diet, right? So right now with the new NRC, what we can do is we can predict intake based on animal characteristics, body weight, body condition score, parity, milk production, right? But we can also contrast that with forage characteristics, right? The ratio ADF to NDF, NDFD, concentration of forage in the diet, and have an idea if what the cow needs can be met with that diet, right? So this is obviously important because we will not only impact milk production, but also the establishment of high forage diets, right? High forage diets, which is one of the strategies that help high producing herds to have good profitability. But the reason why we care so much about forage NDF digestibility is if we go back 20 years, right, or more than 20 years at this point, if we go back to this review from Oben Allen, which was a meta-analysis, they didn't call that during the time, but it's a very similar approach to some of the meta-analysis we have nowadays. And basically what those researchers from Michigan State did, they summarized literature, compile several studies that actually tested for the effects of forage quality on lactation performance. And then they were able to see that for every percentage unit increase in either in vitro or in situ fiber digestibility, animals would consume approximately 0.4 pounds per day more dry matter, right? And obviously, if you think about milk production is driven by dry matter intake. So at the same time, they saw the greater intake when they, with cows fed greater quality forage, they also saw increasing production. More recently, like 10 years ago, Jung and co-authors, they actually run a similar study, but this time they accounted only for diets that contain more than 40% corn silage. And they actually saw a very similar response. And, you know, if you keep digging into literature, into more recent data sets, actually, this continues to be the case, right? So forage quality was a very important concept more than 20 years ago and continues to be that important. But now we know more things about how forage NDF digestibility affect dairy cows. So more recently, we put together a review discussing a little bit of fiber digestibility, forage quality, and animal behavior, especially feeding behavior, to try to understand what are some of those effects. So the data set presented here actually presented data as percentage of controlled treatment. So let me explain that a little bit better now. Okay. So in order to understand fiber digestibility, we decide to contrast diets or experiments that evaluate diets containing either sorghum silage or corn silage. The reason why we chose those is because sorghum silage, except for maybe BMR sorghum, has lower NDF digestibility than corn silage. So we wanted to have this comparison, to have a clean comparison in NDF digestibility. So basically, if we use the first study here, Grant 1994, when you go to the intake column, what this 88.3 means is cows fed sorghum silage consume, sorghum silage-based diets consume 88.3% of cows fed a corn silage-based diet, right? And as you can see, those studies are not necessarily new, and we knew that this was the case, right? So basically intake of cows fed a lower digestibility diet in this case had a dry matter consumption between 85 and 95% of those cows that actually consumed the greater NDF digestibility diet. What we learned during this review was actually the eating time, right? So if we use the Grant 1994 example again, basically what that's meaning is cows that were consuming the sorghum silage-based diet spent 120.7% of the time eating that the cows consuming the corn silage-based diets did. So I think what this data set shows us is that when you feed lower digestibility forages, cows spend more time to eat less, right? So probably there are different things that they are doing there, right? I'm assuming that some of those cows are probably sorting, very likely taking a little bit longer to masticate and consume those diets. So I think it's a very strong evidence that dairy cows are drastically affected by their diet quality. right? So not only in terms of performance, but also because of their feeding behavior. So if we extrapolate intake, performance, and feeding behavior, perhaps some of those cows are spending more time at the feed bunk, less time lying and ruminating, and consequently they are producing less milk, right? However, dairy farmers and dairy nutritionists knows very well that there are several factors that actually affect not only the dairy cow, but also the diet and also the overall performance, so very likely feeding behavior, right? This looks like a crowded slide, but in reality is a very simplistic way of demonstrating that, right? Here are some of the factors that very likely affect feeding behavior, animal intake, and performance, right? So we'll try to dig a little bit in some of those interactions in addition to what we just showed you. So going back some years, we run a meta-analysis to try to understand rumination, okay? And yes, you are not seeing the word rumination in this particular slide, but basically when we run this study, we wanted to understand what changes in rumination would mean for a dairy cow, okay? And somehow we end up learning something completely different, okay? But I'm pointing out that this study was conducted to understand and to learn about rumination, because if you think about that, when we summarize literature, we compile several studies that were designed to manipulate the diet and very likely change rumination, right? Usually when researchers measure rumination is because there's something in the diet that very likely affects that. So there are multiple studies that had different forage quality, different particle size, forage levels, okay? So there are multiple effects here that could be contributing to these results we will discuss next, okay? But during the time that we were running this analysis, we also compiled data related to eating time, okay? The problem with eating time is that we researchers do not know how to properly describe that, okay? And I'm blaming researchers, but I should probably blame myself here, right? Because we didn't have a way to separate what eating time means. The reason why I'm saying that is there are trials that eating time means the time that the cow is truly eating. And there are very likely trials such as those that we conduct here, where eating time is the time that the cow is in our gate feeders, inside the gate feeders, sometimes eating, sometimes sorting, sometimes just there, right? Although she's never just there, right? So eating time here may not necessarily means consuming, okay? You could be doing something else, right? Similar to what we just described about maybe cow sorting, maybe taking longer to masticate, to consume those low quality forage diets, okay? But what is very interesting to me is that this data set was not specifically designed to see forage digestibility. So there are other factors associated with that may play a major role here. And what we saw is that for each minute of increased eating time, there is a decrease not only in dry matter intake, but also in milk production. So probably a lot of those studies, those cows spend more time trying to figure out what they had to eat and less time resting and less time producing milk, right? And yes, this is pure speculation. Probably there is a lot more related to that, okay? Just to give you an example of one of the dietary factors that may change eating time, we have forage NDF of the diet, right? So there is a very strong relationship between the total amount or the total concentration of forage NDF in the diet and eating time, okay? And if you think about studies that evaluate forage concentration, usually when you increase forage NDF in the diet, you have a slight decrease in intake, right? Just because of rumen fuel. So it kind of makes sense, some of those results. Okay. By another hand, rumination also increase, right? So there is a need to find a balance between rumination and eating time, okay? But this study highlighted a lot of things related to the diet, right? Even though we don't know the specifics, of each diet here in this particular meta-analysis. This is primarily related to diet, and there is much more to that, right? Nutritional management plays a major role. The animal characteristics play a major role. So last year, we were funded a study by the Dairy Innovation Hub to work with a data set from previous studies conducted using these blue gates you can see in the picture here, which are the gate feeders we have in our dairy research facility in Arlington. And basically, the goal we have multiple goals with this study, okay? One of those is to understand UNDF intake, which I will not be showing today. We are summarizing the data. But a second goal of this study is to understand the relationship between feeding behavior and lactation performance. And that's some of the data that I will present and discuss with you today, okay? So just so you understand, there are multiple ways of conducting studies using this particular set of gate feeders. So basically we have a pen that we can have 64 cows at a time, and there are 32 of those blue feeders. So the studies that we use to understand feeding behavior, cows had access to any feeder, okay? We didn't restrict those cows, okay? Sometimes when you run nutrition studies, because we test different diets, cows only have access to gates that have their assigned diets, okay? I'm just mentioning that so you fully understand what we will present. The way those gates work is those cows have an RFID tag in the ear, and then when they put their head inside the gate, if those gates allow them to enter, the gate open and they enter. In this case, because they could enter in any gate, the gate would open, okay? But more important, when they do that, the gate record which cow is there, and because those have weight cells under those feed bunks, we can record individual intake and later calculate all sorts of feeding behavior. Okay. So basically, we try to repeat the same metric we just used with that literature data set, right? We try to understand the effect of eating time on lactation performance. But in this particular case, cows were consuming either the same diet, because they were all together in that particular pen, or they were consuming a very similar diet, because actually those studies that we use don't differ a lot in diets, right? Maybe change a little bit the time of the year, so it changes slightly the forage quality, but overall the diet didn't change much, okay? So pretty much what we are testing with this data set is the variation within cows, okay? Animal characteristics, effects on eating time. So you can see that our results are actually very different than what we saw when we did that literature search or literature review and meta-analysis. We did not find any relationship between eating time and intake, milk production, or fat yield, right? We saw a minor change in protein yield, but compared with the previous literature trial, is actually very, very different, right? Well, Luis, so what do you think of that, right? Well, what I think is that when we consider feeding behavior, we should consider multiple factors, right? Previously, we were trying to understand the diet. Now we are seeing some effects related to the animal, and very likely there are multiple factors associated with how we manage a given pen or a given herd that change that as well, okay? But then we decide to dig into a couple new things. And something that's very unique about the system is that because we know the individual intake and the time that those cows spend eating, we could calculate eating rate, okay? Eating rate is the amount of food that the cow consumes per minute, okay? Basically per minute of eating time, okay? In this particular case, please don't pay too much attention to the effect number here, as basically eating rate in our case is calculated as kilograms per minute. And here we are trying to show you what is the effect of each kilogram per minute greater eating rate on lactation performance. There's just one problem with that, okay? Eating rate usually for our cows goes between 0.1 to 0.25 kilograms per minute, okay? So one kilogram change is too much, okay? We just put those here so you have an understanding of what is going on, okay? How does that change? So basically, cows that consume more per minute, they have greater intake and greater milk production, right? And obviously, greater milk component yields, right? And I think this is a very interesting finding because, you know, in general, when we think about greater eating rate, usually are diets that are either more susceptible to acidosis, like high concentrate diets, right? Or we think about cows that that are doing slug feeding, right? In this particular case, cows are within a normal range of eating rate. So we are comparing normal cows that are not really slug feeding, okay? So if you have a normal management and your management does not induce slug feeding, the eating rate is probably something that is individual of each cow, and the cows that are able to eat faster looks like they can produce more, okay? But then we start digging a little bit more on that and try to understand what does that actually mean, right? What else is affecting those feeding behavior patterns, right? Instead of performance, to try to figure out if there is something there that we can learn and maybe use later on for management practice, right? So the first thing that we decided to go through was parity. And before we go through some of the data, I just want to mention that as we reach the older cows, like 6, 7, 8 lactation, we don't have a lot of cows in this data set, right? You can see that for the 8 lactations, we only have 1 cow, right? A very good cow, but only 1 cow, okay? And then for 7 lactation, we have 2, and for 6, only 8, right? In contrast, we have 85 second lactation animals, okay? So be careful because of the low number of animals that we have for some of those parities, okay? But something that I think is really neat when comparing those different parities is that the older cows, they eat more and produce more, okay? But they spend less time eating. And I think that's actually really nice and it makes a lot of sense to me, right? They go there, they eat, and then they go to rest, right? And pretty much they have my behavior. They like going to eat and then resting, and it works very well, right? But when you go to their eating rates, because they eat more and spend less time eating, they actually have a much faster eating rate, right? And if you calculate their largest meal in a day, actually they have a largest meal than the younger cows, right? So, well, Luis, what does that mean, right? Well, it could mean a lot of different things. Maybe that older cows should be grouped together. I'm not saying you necessarily have to do that, but maybe this may have other implications from a day-to-day nutritional management. Another very interesting thing to me is that those cows actually visit the feed bunk. If you get the 7 and 8 lactation cows here, and yes, 3 cows only, but they visit the feed bunk 10 times less, than the second lactation cows, right? Which goes a lot to different feed bunks, right? Probably there is another behavior component there that we cannot dig with this specific data set, right? Maybe some dominance or maybe some displacements that we don't see, but I think it's a really, really nice data set. The cows eat more, produce more, have greater eating rate, but spend less time and visit less times the feed bunk, right? So, but now I want to show another part of this data set and how this translates into the day-to-day management, okay? But before doing that, I just want to introduce this concept of TMR consistency, okay? So obviously this is a scenario that probably many of you saw before, right? And here's farm 1. And basically TMR consistent in this particular case was measured as 10 samples collected from the same TMR across a feed bunk. And then number 11 is the average of those 10, okay? And then they used the Penn State shaker box, the set of sieves, to measure particle size and see if the particle size of the diet is consistent across the entire feed bunk, right? And if you think about that, it looks like a very, very consistent delivery of TMR, right? The TMR looks pretty similar across the entire feedbunk, okay? Then let's talk about your neighbor next, right? So that's your neighbor, and basically your neighbor went there and fed their cows, and you did exactly the same thing. 1 to 10 are the different spots across the feedbunk. 11 is the average across all of those. Well, this is not very consistent, right? There is a lot of oscillation, right? So I guess the question is, if you are a cow and you arrive there to eat and you like the spot number 1, and then after that you go to spot number 5, those are not the same meals, right? They're very likely different, right? And just for the record, this is collected at delivery. So the cows didn't have time to sort yet. Okay, we'll discuss sorting in a little bit, but just to give you that perspective. Well, why is that important, right? Cows theoretically can sort for what they want to eat. They will walk across the pen and, try to figure out what to do. You're right, they do that. However, there is something that has to be considered, right? So this is a survey conducted in Canada, and 22 dairy farms were visited for 7 consecutive days over the summer, and then after that, over the winter. And basically, the researchers collected samples and measured particle size to calculate variation. Okay, CV is a coefficient of variation. It's a metric that we use a lot in the lab, right? Basically, what we create thresholds to figure out what is a good variation, right? So for example, sometimes we're measuring samples here in the lab, and if CV is greater than 5%, we run the sample again because it's not good enough. We don't know if the result is right? In this case, we are just seeing consistency. And what those researchers learned with this specific survey was that farms with less variability were associated with greater intake and greater milk production, right? Another thing that they learned with this survey related to TMR consistency is that actually there is more variability if they contrast the nutrient composition of the formulated diet to the offered diet than the day-to-day variation in nutrient composition, okay? However, if you go through this table and the different particle size, you see that there is a good amount of variation in the large particles, right? As well as the bottom pen, which means that probably there is some fluctuation in the proportional forage of that diet day to day, okay? So, and in this particular case, also across the feedbunk, right? So probably there is a mixing issue there, or sometimes an amount issue there across those farms, right? Well, what does that mean to the dairy cow, right? So many years ago, Canadian researchers evaluated what is the preferred spot for heifers, right? And they learned that heifers actually have a preferred portion of the feedbunk. And they learned that diets that were inconsistent, similar to those that we show from that particular survey, actually increase the amount of times that they change their eating spot by more than 50%, right? Because they don't want to eat certain diets, they want consistency, right? Cows have a routine, right? So basically, inconsistent diets cause 3.5 more bunk displacements and changes in their eating spot compared to consistent diets, right? So that plays a major role. So we decide to check, does our data set give us any hints about the behavior of cows instead of heifers? And there is a lot of information in this graph, okay? But just to go through, okay? For this set of cows, we calculated the proportion of intake of those cows from each of those 32 gate feeders, or each of those 32 bins, okay, for their entire experimental period, in this case, 42 days. So there is data from 42 days here across all gates, okay? And what I wanted to show you is the cows differ a lot, right? So if we go through some of those, if I use 8180, for example, okay, which is right in the middle here, you see that this particular cow does not have a good proportion of their meals on the left side of the pen. So I can assume based on her data that the gates 1 to 15 or 1 to 16 are very likely not their favorites. Okay, so she doesn't like going there for whatever reason. Okay, so if I don't have a consistent diet and whatever I'm delivering from gate 16 to 32 or 17 to 32 is not good, we'll have problems, right? And if we start going through all those, you see that, you know, to different extents, those cows differ on where and which gates they like to eat or having some of their larger meals, right? And I think this is very important because if we go back to that TMR consistency discussion, if she goes to her favorite spot and the meal is not really consistent, either she's gonna eat something that is very different than what she expected and you as a nutritionist or dairy farmer is expected to offer, or she's gonna look for another spot. If she looks for another spot, she probably will try to take another cow that may be eating on her specific or favorite spot, and then we start creating displacements, and then we start creating competition, and then we start creating problems. Competition leads to slug feeding leads to a lot of other issues, right? So I think we are still evaluating this data set, but I thought it was very nice to visualize some of those preferences, because it's very obvious that some cows prefer the right side over the left and vice versa, and some cows prefer to eat in the middle. Probably after they establish dominance and get acquainted with each other, they probably create their own spots and go from there. So, okay. And going back to the discussion, why is this important, right? Not only because of TMR consistency, but also because of sorting, right? So if a cow has a preferred spot, and in our case, it doesn't matter because we don't have push-ups and those gate feeders, but if there's a lot of sorting in a spot, and then you don't have enough push-ups to move those back and make the diet a little bit more similar to what's delivered, then you may have the same issues, right? And just to show some data on that, the same survey, okay, published in a different article, but the same survey from Canada, they actually measured that. They offer and refuse diets from these 22 dairy farms, 7 consecutive days over the summer, 7 consecutive days over the winter. And what they saw is that dairy cows sort for smaller particles, right? They refuse the large particles. They refuse some of the physically effective NDF, right? And the physically effective NDF is there for a reason, right? Rumen health, make sure cow will produce enough milk fat, and so on. And last, I want to finish discussing one last interaction between diet and management, okay? I always sell the idea of how important fiber digestibility is, not only for performance, but also for feeding behavior, as I saw today. But probably there are scenarios where having a less digestible diet may be important too, okay? So this is a study conducted at the Miner Institute, and they were trying to understand the interaction between overcrowding and diet characteristics. Right? In this particular case, they had 2 stocking densities, 100% and 142. And then they had a diet containing 9.7% UNDF or 8.5% UNDF. Those were cannulated cows, and they could measure rumen pH over time. So basically what they learned is that the cows that were in those pens containing 100% of stocking density, their rumen pH was below 5.8, approximately 2 hours per day, okay? Why 5.8? 5.8 is the threshold that we use for subacute ruminal acidosis, okay? Just to give us an idea of that. However, cows that were in those pens with 142 stocking density, if they were consuming the more digestible diet, they stayed twice the time under that threshold. Okay, so the reason why I'm bringing this slide and this specific information is perhaps there are scenarios where you cannot control stocking density and may be worth considering a slightly less digestible diet because of slug feeding to avoid drastic drops in rumen pH and consequently milk fat depression and maybe some other issues. So with that, there are many factors that alter fiber digestibility of forages. We didn't discuss some of those, but we discussed some of those indicators related to that. More digestible forage sources increase intake and modulate feeding behavior patterns. However, there are multiple animal and feeding environment characteristics that alter feeding behavior and lactation performance. And at the end of the day, those should all be considered together to provide, I'm gonna say, the perfect diet for those cows. With that, I would like to thank you for attending this webinar. And if there is any question, I will be happy to answer.
All righty, thank you, Luiz. Again, if you do have any quick questions, you can feel free to drop them in the chat box.
So Luiz, the first question you have is, have you done any work looking at cow performance and cow intake in cows which spend part of their time grazing?
I have not. Our facilities are primarily designed for full TMRs, and we don't have specific areas to combine those. But you know, if we go back some years before moving to US, I actually spent 6 months in a co-op in Brazil, and a lot of the dairies there, small scale dairies, low producing cow dairies, they actually had this system. And although we cannot compare performance, we can have an idea on intake and some of these feeding behavior patterns, similar to what we see with those lower digestibility diets, as well as feed availability for cows fed TMRs. If we have low quality pasture or poor managed pastures, as well as low availability, when those cows reach in that case, they used to be fed right after milking, right? A partial TMR. So those cows that consume low quality feeds or low quality forages, when they reached that stage, it actually created a lot of competition in eating time right after milking, right? So I think that the behavior resembles what the TMR fed cows do, but unfortunately I don't have specific numbers on intake and performance that would be comparable to the conditions here in the US.
I don't see any other questions.
And if there are not any more questions, we'd like to thank everyone for joining us today on our bi-weekly Badger Dairy Insight program. In 2 weeks, we will be hosting the Animal Well-Being Conference, and I backed up the slides, the Wisconsin Dairy and Beef Animal Well-Being Conference, which will be held over in Manitowoc at the Farm Wisconsin Discovery Center. If you are unable to make it in person, you can register for the virtual option as well. So please do that. Otherwise, we will be back for our winter series, the Badger Dairy Insight, March 1st, talking about the ins and outs of cocktail forage mixes for dairy rations. So again, we'd like to thank everyone for coming today. Please answer the poll questions, and we will see you either February 22nd or back here on the Badger Dairy Insight, March 1st. Thank you.